Welcome to Biotechnology World. Now our topic molecular docking. Introduction to molecular docking. 1. Molecular docking is a computational technique used in drug discovery to predict the preferred orientation of one molecule to a second when bound to each other to form a stable complex. 2. It aids in understanding the interaction between small molecules, that is ligands, and target macromolecules, receptors such as proteins or nucleic acids. Objectives of molecular docking. 1. The primary objective is to predict the binding mode and affinity of a small molecule to a target protein, which can provide insights for drug design and lead optimization. 2. It helps in identifying potential drug candidates by assessing the likelihood of binding to the target protein and forming stable complexes. Major steps involved in molecular docking. 1. Preparation. Preparation of ligands and receptors involves cleaning and optimizing the structures to ensure accuracy in docking calculations. 2. Search algorithm. Various algorithms are employed to explore the conformational space and find the optimal binding pose of the ligand within the binding site of the receptor. 3. Scoring function. Scoring functions are used to evaluate and rank the binding pose generated by the search algorithm based on the predicted binding affinity. 4. Validation. Docking results are validated using experimental data or through cross-validation techniques to ensure reliability and accuracy. Major types of docking. 1. Rigid docking assumes that both ligand and receptor maintain fixed conformations during the binding process. 2. Flexible docking accounts for flexibility in both ligand and receptor, allowing conformational changes upon binding. 3. Virtual screening. High throughput docking used to screen large libraries of compounds to identify potential lead molecules for drug development. Applications. 1. Drug discovery. Identifying novel lead compounds and optimizing their binding affinity and specificity for target proteins. 2. Structure-based drug design. Guiding the design of new drugs by understanding the molecular interactions between ligands and receptors. 3. Predicting binding affinity. Estimating the strength of binding between ligands and receptors to prioritize drug candidates. Challenges and limitations of molecular docking. 1. Scoring accuracy. Scoring functions may not always accurately predict binding affinities due to the complexity of molecular interactions. 2. Flexibility. Handling flexibility in both ligands and receptors increases computational complexity and may require advanced algorithms. 3. Validation. Validating docking results experimentally can be challenging and time consuming, especially for large-scale virtual screening campaigns. Future directions about molecular docking. 1. Integration of experimental data. Incorporating experimental data such as protein-ligand interaction kinetics and thermodynamics to improve docking accuracy. 2. Machine learning approaches. Utilizing machine learning techniques to enhance scoring functions and predict binding affinities more accurately. 3. Multi-target docking. Expanding docking methods to consider the interactions of ligands with multiple protein targets simultaneously for polypharmacology studies. Thank you.